DNA is antiparallel, so it's 5 to 3 to 3 to 5 prime. DNA helicase unravels the DNA. Single-stranding binding proteins keep the DNA from getting back together. Primase sets an RNA primer on the DNA. DNA polymerase constructs a new DNA strand from the RNA primer. The exonuclease removes the primer that was given to by the primase. Finally, ligase replaces the missing strand with a new strand and makes sure everything is stuck together in the DNA. In the leading strand, the primer is only needed once or twice. DNA polymerase can't move from 3' to 5', so this is the lagging strand. The lagging strand is where the primase is needed much more often to create individual primers so that the polymerase can make new strands of DNA. When the primers are set and the DNA polymerase does its work, this creates Okazaki fragments. Okazaki fragments are short, newly synthesized DNA fragments. Finally, the exonuclease removes the primers and the ligase tidies everything up. It fixes all the missing areas, and that's DNA replication. Another fun fact, topoisomerase is an enzyme that alters the supercoiled version of the DNA molecule so that the helicase can break apart the DNA strands to allow DNA replication. Also, DNA polymerase 3 is essential for the replication of the leading and lagging strands, whereas DNA polymerase 1 is essential just for removing the RNA primers from the fragments and replacing it with the required nucleotides. These enzymes cannot work without each other. DNA polymerase can only proceed from 5' to 3', and the lagging strand on the DNA is discontinuous because it goes from 3' to 5', and the polymerase can't do that with just one RNA primer. So the primase has to make multiple primers so the polymerase can follow itself till it reaches the other side of the origin. The energy to create growing strands is coming from the nucleoside triphosphate groups when they are taken from the polymerase and put into the strands. The ATP is used to create the growing ones.